At the outset, I would like to thank the organizers for inviting me over. Today, I will be talking about how inflammation is an important component of the tumor microenvironment, and I'll be talking in particularly about the glioblastoma, which happens to be one of the most malignant of brain tumors. Now, when we envisage a tumor, we think of it as a homogeneous mass of tissue, but in reality, it is the cancer cell, the proliferating cancer cell in a tumor environment that is, bathed by the immune cell. They are the fibroblasts, they are the endothelial cells, which are the co-conspirators in the tumor progression. So they support the proliferation and the progression of tumors. So therefore, the tumor microenvironment is a very complex one, and therefore is a very good target for chemoprevention. And cytokine-mediated link between cancer and inflammation has been well established over the last decade, and various groups have shown that inflammation, the tumor microenvironment, is an important component in the disease progression. Now, this is the hallmark that you are all aware of, and which Dr. Kundu has already shown you. The hallmarks of a cancer cell, like its ability to sustain proliferative signals, resist cell death, replicative immortality. But what has come of recently are the emerging hallmarks of a cancer cell, whereby avoiding immune destruction, the tumor promoting inflammation, and deregulating cellular energetics are considered the most important component, in addition to the ones that you have seen, in regulating the enabling characteristics of a tumor. Now, I shall be talking about the tumor promoting inflammation, as well as how this inflammatory environment enables a tumor to avoid immune recognition. Now, talking about inflammation, you will agree with me from this slide that though I am talking about several cytokines and the chemokines that are involved in the tumor microenvironment, they kind of narrow down only to a very few transcription factors. So even if we start with a plethora of cytokine here, if you see that we have nailed down to a few very common transcription factors like STATs, NF-kappa B, and therefore, if I could put it in another way, there are many more signaling pathways that you can see here than are the oncogenic transcription factors. Importantly, therefore, if you listen to many of the speakers over here today, you will see there are only few transcription factors that are repeatedly coming up in your conversation: NF-kappa B, HAPE, STAT. So you will agree with me that there are limited transcription factors that are overacting in most human cancer. Therefore, they are the most direct and the hopeful targets for treating cancer, given the limited numbers. Now, talking about glioma, as I mentioned, they are the most malignant of brain tumors, and they're highly heterogeneous. So that's the reason they derive the term multiforme. It's astrocytes, there are stem cells which are present in the fibroblasts. It is multiforme, it is heterogeneous. But the worst thing about GBMs, they are highly resistant to classical therapies, including surgeries, irradiation, chemotherapy, and have a very poor prognosis. Gliomas have constitutively highly active level of NF-kappa B, and as Padma Shastri has mentioned in a talk, the tumor necrosis factor is a transducer of NF-kappa B, and what happens at glioblastoma are resistant to TNF-induced apoptosis. Now, from the slide, you can see that TNF can induce both cell proliferation as well as apoptosis. But in GBM, what happens, it sustains the cell proliferative ability through induction of NF-kappa B, and this is additive of the already highly constitutive levels of NF-kappa B present in the glioma cell. So GBM are resistant to NF-kappa B, to TNF-induced apoptosis because of the high levels of NF-kappa B that is already present. I'll be talking about another transcription factor which my lab has been working for the last 5 years. Is called the hypoxia-inducible factor, and which Dr. Kundu has already mentioned. It is a transcription factor which is upregulated in response to low oxygen, that is to hypoxia. Under normoxia, HIF is present, hypoxia-inducible factor, HIF. HIF is rapidly degraded. But under hypoxia, what happens, it is stabilized, and then HIF dimerizes with its partner, HIF1 beta, and together they go and bind to the HIF responsive elements, that is HREs, which are present on the promoters of target genes which HIF regulates. And what are these target genes that HIF regulates? They are the VEGF, they are COX-2, IL-1 and MMPs, genes that are associated with tumor progression. Now, with the previous slide we have seen that hypoxia induces HIF. And in our lab we found that in glioma patient samples there is a highly elevated level of interleukin-1b. And therefore we question, constitutive NF-kappa B is high in this tumor, there is a high level of pro-inflammatory cytokine, and it is known that NF-kappa B drives HIF activation. So the question we ask, does inflammation has anything to do with induction of HIF? So what we found that IL-1 actually can drive HIF1 transcriptional activation through RAS oncogene. So this was the first time we are showing that in addition to hypoxia, an inflammatory microenvironment can also drive the transcriptional regulation of HIF. But the most important thing that we've seen that this HIF goes and binds to the HIF responsive elements upstream of the IL-1 promoter. And what it does, it again puts on the vicious cycle of generating more inflammatory cytokine. So this is the reason probably we find about 1,000-fold high levels of IL-1 in the GBM tumor sample as compared with the normal tissue. So this maintains a necessary cyclicity, if I could put, of sustaining a high level of a tumor pro-inflammatory microenvironment. Now, toll-like receptors, as Dr. Shastri has mentioned, they are associated with innate immune responses, and they are usually PAMs with a pathogen associated. They recognize molecular patterns. But interestingly, what we found out, because why we got interested into TLR4 is that TNF and TLR4 shares common signaling pathways, and what we found that TNF can actually, through TLR4, TNF regulates TLR4, which induces HIF1 activation as well as NF-kappa B activation, and this TLR4 maintains the feed-forward loop, which again goes on and sustains the inflammatory response in the cell. But what turned out to be very interesting when we found that HIF can actually regulate true crucial component of the microenvironment. One is the growth factor, insulin-like growth factor, and another is cytokine. So basically what I'm trying to say here, HIF is at the crossroad of inflammation and tumorigenesis through regulation of toll-like receptors associated with innate immune responses, or through RAS, which is an oncogenic RAS which plays an important role in transformation. At this point, when we were looking into the expression of cytokines, chemokines, Devrat, a grad student, he looked into the expression of casein kinase 2 in these tumors, And what he found that casein kinase 2, by the way, it is ubiquitously expressed in tumors, is a kinase and which regulates NF-kappa B activation. What we found that alongside the high activation of casein kinase 2 in GBM, grade 4 we are talking about, there is also an increased expression of SIRT1 and HDAC. Through a series of experiments, what Devra showed that when you inhibit casein kinase 2, it inhibits NF-kappa B activation, but what he also seen that it regulates p53 activation through sirtuins. So we are not only showing that regulation of NF-kappa B through casein kinase 2, but we are actually showing a regulation of p53 to HDAC sirtuin. So with that, given the importance of NF-kappa B in the environment, and that besides chemotherapy, given that radiation is also an important way of treating GBM, we wanted to see what happens upon treatment with ionizing radiation in this cells, and because the reason being that radiation-induced NF-kappa B mediates radiation-resistant in glioma, it's known. Resistance of GBM to TNF-induced apoptosis is contributed to NF-kappa B, as I've already said. And NF-kappa B TNF crosstalks promote survival following radiation. Now with that, we wanted to see what is the status of NF-kappa B in cells treated with a radiomimetic, both in the presence and in the absence of TNF, because TNF, gliomas are resistant to TNF. But what we see here, so what we wanted to really address is, does inflammation affect the responsiveness of glioma cells to radiomimetic? So where it shifts, where inflammation plays an important role in tilting the balance between death and radioadaptation, whether it leads to more radioadaptation, radioresistance, what is it? So it came as a surprise to us when we found that in the this is neocarcinostatin N, and this is T plus and TNF. What we find that with TNF there is no death, obviously, but neocarcinostatin sensitizes glioma cell to TNF-induced death, but we find an increase in NF-kappa B activation despite death. This was accompanied by, and what we found that when you inhibit the NF-kappa B activation, there is more neocarcinostatin inducing death. This implies that the level of NF-kappa B that is being induced in the presence of an inflammatory microenvironment, it is preventing the fullest manifestation of the death-inducing potential of your radiomimetic. Same is the case with NF-kappa with AKT, which is a pro-survival marker in glioma, is highly activated, is pro-survival. And inhibition of AKT, what happens, it further reduces, it enhances the ability of neocarcinostatin to induce death. Importantly, what we see, surprising, that in an inflammatory environment, when we give radiomimetics, there is an induction of HIV. So if I could summarize this part, I would say that neocarcinostatin alone has no effect on NF-kappa B, and but in an inflammatory environment, what you see that there is a complex regulation of pro and anti-survival mediators, because AKT, ERK, NF-kappa B, HIV, which is tumor promoting, gets induced in the presence of an inflammatory microenvironment, in the presence of radiomimetic. There was an upregulation of certain metabolic genes associated with metabolism. So what I want to state here, that inflammation affects resistance and adaptation to radiation despite concurrent induction of death. So there is death, no doubt, but there are certain cells that become resistant, that survive in this inflammatory environment. Talking about apoptosis, we are not only concerned with resistance to chemotherapy, apoptosis to in the presence of radiation, but also what happens in what is the status of the immune escape mechanism in this cell. Does inflammation affect immune escape? Because gliomas have effective immune escape mechanism, but gliomas express high levels of MHC class 1 molecules. And what does that do? It makes them resistant to natural killer cell mediated cytolysis. So, and there are reports that a hypoxic tumor microenvironment contributes to immune escape mechanisms in cancer. So we wanted to see what is this apparent paradox. There is high levels of MHC, but still the cells are not able to, evade, they are able to evade immune response. And why, from where is this high level of MHC, what is triggering this high level of MHC? What we see here, we are talking here about the classical MHC ABC. We find that both hypoxia as well as TNF induces MHC class 1 molecule. This is accompanied by the induction of beta-2M, and because MHC has to, form a complex with beta-2M where the peptide, that is a different story, but what I'm trying to emphasize is that hypoxia plays an important role in the regulation of MHC1, and this occurs very much in a HIV-dependent manner. This was a classical MHC1 molecule. Then there are the non-classical MHC molecule, which is HLA-G, and through a series of experiments, we found that human beta defensin, which is associated with adaptive and innate immune response, and HMGB1, which mediates immune suppression, they are highly elevated in GBM. And what Piyushi, who did this experiment, she found there is a complex signaling that goes on whereby IL-1 regulates TLR4, HLA-G, and HMGB1 through HIF, and beta defensin kind of negatively regulates these circuitries. So we have actually nailed down to two axes in GBM, like the beta defensin TLR HLA-G and TLR HLA HMG1, that works in tandem to regulate the level of HLA-G in the cells. So the take-home message, if I could consolidate whatever I've said in this one slide, I could say that therefore transcription factors play an important role in, in modulating how an inflammatory microenvironment will dictate the progression of a tumor, and therefore targeting this transcription factor may be an open frontier for glioma therapy. But that doesn't necessarily mean that these only transcription factors are important, because to me, as the origin of species, is by means of natural selection, the same kind of holds true for a glioma. It's a tumor microenvironment. The question I'm putting forth is the neoplastic tissue therefore a Darwinian microenvironment, whereby the microenvironment gradually sculpts the tumor cell, gradually sculpts the the environment, whereby it selects a particular type that fits better into the environment, and it kinds of there's a natural selection that goes on that kind of selects and fuse together the different variants that the immune system has sculpted, or the inflammatory microenvironment has sculpted, to give rise to specific population that are most fit to survive in the deleterious environment. But as the quest goes on, the quest to find the relation between inflammation and glioma progression, I would like to acknowledge the contribution from the students, the present and the former student, and my postdoc over here. And at the end, I would like to thank you all, but leave you with this question that the methods of nature are really very complex, and who are we to ever analyze it? And with that, I would be very happy to take in any questions, and thank you very much.